Mahalia. Yeah, so Donald Tweedy, I'm an ADME lead scientist within the PPDM organization at Merck, and I've been involved with the induction working group since its inception. My interest is ensuring that we have effective interactions with regulators to drive towards a scientifically defensible position on drug-drug interactions. And this entails both an understanding of the perspective of regulators and also pushing for a pragmatic industry perspective to minimize the need for studies that don't add value, but also ensure that we maintain patient safety. Our presenters today are Jane Kenny, who's an Associate Director and Principal Scientist in DMPK at Genentech. She received her PhD from the University of Liverpool and has over 15 years of drug discovery and development experience. Jane led the data interpretation subgroup for the IQ induction working group, which is the main topic of today's presentation, and was a manuscript submitted to published in DMD earlier this year. The other presenter is Diane Ramsden, who's a senior scientist in early development at Alnylam Pharmaceuticals. She received her BS from the University of Connecticut. She's currently responsible for representing drug metabolism and pharmacokinetics on program teams and for designing mechanistic ADME studies. Prior to joining Alnylam, she spent 16 years at Boehringer Ingelheim with me, and where she worked as a DMPK project rep and was our hepatocyte lab head and our go-to person for induction studies. As well as being a core member of the induction working group, she's a lead author on a commentary which is being prepared on variability in clinical studies, where she really synthesized a large amount of data from the literature. We also have Dr. Naresh Hariprasad, who will be a panelist for the Q&A and will, with me, be leading or asking the questions that you submit. Naresh is a director of Vertex Pharmaceuticals. He received his B.Pharm and Master's in Medical Science from the University of KwaZulu-Natal in South Africa, and a PhD from the University of Cincinnati. And he's been at Vertex since 2010. And he's functioned as the chair of the induction working group since it started in 2014. So, the genesis of this induction working group was really a Q&A session at the Toronto International ISSX meeting, where industry was providing very specific feedback to a presentation from the EMA. When questioned, Eva Gill-Berglund was very receptive to the proposal that industry provide data to support specific alternative recommendations on the DDI guidance, specifically induction. And so that induction working group derived from the IQ Drug Metabolism Leadership Group, which is now the Translational and ADME Sciences Leadership Group, with the goal of providing a data-driven approach to understand specific aspects of DDI guidance. A short list of specific feedback was compiled, some of which was addressed in the first webinar and is the first white paper in DMD. Jane will introduce today's topic, and there are two additional publications which will be talked about at the end of the presentation from the induction working group. One on a time course for in vitro induction and data analysis, and the other is the commentary that I previously mentioned. We encourage feedback, both positive and negative, and so please, whether through today's Q&A or through feedback directly, there will be email addresses at the end of the presentation. Please send your thoughts on today's presentation or on any of the induction working group publications. And I'll now hand it over to Jane to start the presentation. Thank you.
Good morning, good afternoon, everybody. It's my great pleasure to represent the induction working group this morning. And we're going to be talking about the findings on the CYP3A4 mRNA in vitro response thresholds, variability, and clinical relevance. Can you advance the slides for me? It's not working, Mahalia. Sorry. Thank you. So to set the stage, in addition to what Donald has introduced, the induction working group had two major objectives. The first to move to a better understanding of the overall regulatory approach for induction-based DDIs, and the second to then take a data-driven approach to work with regulatory agencies to update potential requirements for assessing induction. And as mentioned, this was published in our first manuscript last year, and Naresh presented this at the webinar that I hope many of you were able to attend last week. There were 5 areas identified. Downregulation, 2C induction, and 2B6 positive control had been addressed. The other 2 were data interpretation, which we'll talk about today, and time course of induction, which is going to be a future output from the induction working group. Next slide, please. Thank you. So for the induction working group, subgroup on data interpretation, we were working pretty intensely over the last 4 years to address a primary goal around better defining in vitro induction positive and negative cutoffs. And we did this using a collection of data that we pulled from multiple different companies and from the literature. And we focused on the appropriateness of a 2-fold mRNA threshold. And this is what's been particularly recommended in the EMA guidances. Next, please. So there was a large group of people, and you can see some of us here. And we represent a diverse range of companies. And not everybody who presents, who was part of the manuscript, unfortunately, is represented in this picture. But you can see that there was a lot of people collaborating on this work, and we're going to talk about the outputs now. And there were 6 recommendations published in the manuscript that was in DMD earlier this year, and I'll talk you through them in some detail. The first was that the CYP inductions should continue to be evaluated in 3 separate donors. The second is that with the variability we're seeing, normalization of data to the % positive control doesn't really help us control for that in vitro variability. Next. Then 2-fold induction is actually an acceptable threshold for positive identification of 3A4 mRNA. I think many of us, myself included, felt that 2-fold was very conservative. We were thinking that once we gathered a lot of data, we would be able to show that this was a very conservative threshold and we would be able to raise that. That is not the case, and we'll talk through the data today. However, we're saying 2-fold is appropriate, but the next recommendation is that to reduce the risk of false positives, we need to see 2-fold in more than 1 donor in order to classify a compound as an in vitro inducer. Next, if you're qualifying a compound as a negative, it's important to ensure you have sufficient dynamic range in your assay. And we're saying that we recommend rifampicin, your positive control, should be at least 10-fold in that donor if you want to qualify less than 2-fold as a negative for in vitro induction. And finally, the inclusion of a negative control is not adding value beyond the vehicle control that is already in the system. The majority of the presentation that I'm giving this morning is going to focus around these recommendations and the data that supports them. And some of these are in support of the guidances. I'm highlighting that there in green. So 3 donors, 2-fold threshold is in line with the regulatory perspective right now. However, there are several layers of new practice that are introduced and recommended by the induction working group. And we'll talk through those in detail. And those are normalization of data to % positive control isn't really helping us. You should see greater than 2-fold in more than 1 donor if you're qualifying a positive. And you should have sufficient dynamic range and 10-fold for rifampicin. I'm listing 1 as confirmatory, and that's the negative control. Now, whilst this is still a practice this is still recommended in the guidances, I think we found in practice in talking to many different organizations, not too many of us are actually including a negative control. And actually the data that I will show you supports that, and that's why I've listed this as confirmatory. So let's get into the data, and we're going to start with clinical data, because everything we're doing in the in vitro setting is to try and assess a clinical risk. So we started from that point, and we gathered a lot of data. We have clinical induction data for 51 compounds. 16 of those were proprietary, so came from IQ member companies, And this is data that is not publicly available and just shows you the power of this sort of consortium work. 35 compounds were gathered from the literature, and we focused on the mild or moderate induction. So this is not an extensive list of every compound that has published induction DDI. We're really focusing on that tricky range to predict, the mild or the moderate. We're pretty comfortable that we can identify strong clinical induction. To describe this data, I'm showing you two sets of figures. The one on the left is the number of compounds, and I'm defining the category of drug interaction based on a series of colors, strong being less than 0.2 AUCR ratio. So this is the ratio of the victim drug in the presence and absence of the perpetrator. And then orange is moderate, yellow is mild, and green is no drug-drug interaction. And you can see from these two panels how you actually look at your clinical data does drive the category somewhat. And Diane will talk about this in more detail later on. But on the left panel, we're classifying according to the worst case or the strongest induction study in the cases where there are multiple DDI studies run for a single compound. If we use the median of those clinical studies, you can see the categories do shift a little bit. And I'm drawing attention to this because when we think about translating our in vitro data to a clinical meaningful prediction, the clinical target is somewhat moving. And so we have to be mindful about what we're actually trying to predict. Are we trying to predict the average interaction in the clinic? Or are we trying to predict the worst interaction that we could have? And that really would drive how you go about doing your IVIVE. So Diane will talk more about the IVIVE in the latter half of this presentation. I'm going to show you more clinical data now. So this is the breakdown of the compounds that I just described, those 51 compounds. I'm showing the actual AUCR ratios, and the data set is divided into two parts. On the left, this is compounds where the in vitro data showed induction only, and then on the right, where the in vitro data showed induction and inhibition. And this inhibition could be reversible or time-dependent inhibition. A couple of striking observations when you look at this data. First is the degree of variability that's observed. If you look, for example, for rifampicin, you can see quite a wide range in the clinical induction that was observed. The other thing that's striking is that there's a large number of compounds that have mixed mechanism. This panel on the right is over 60% of our data set, which means that predicting the DDI that actually predominates in the clinic is not straightforward. Okay, I'm now going to go and talk into a little bit more depth on the actual in vitro data. So next slide, please. And I want to start by focusing on weak in vitro induction. Because this is really the area that this group focused on. And I'm starting by telling you that even weak induction can have clinically significant DDIs. And this is important when we context that twofold threshold for CYP3A4 mRNA. And I'm showing data for 10 compounds here where the Emax at the CYP3A4 mRNA is 3 or less in at least one of those donors. And so let's look at compound 7 at the bottom as an example. And here you can see that there was 3-fold to 3.1 to 4.6, and there was 3 different donors run. The median is 3.4. You can then contrast these compounds that had at least one of the donors with a very low response to their clinical DDI, which I'm showing in the blue panels. And importantly, if you can advance one more click, you can see that for compounds like pleconaril and compound 7, there are clinically significant DDIs despite the fact that there is very weak in vitro induction. So only 3-fold in vitro led to some significant drug interactions. And this highlights 2 things. One, the importance of that 2-fold threshold if 3-fold is able to cause a DDI, and also that we really need to be able to detect weak inducers in our systems. Next, please. Now, of course, it's not just the Emax that drives whether you're going to see an interaction. The EC50 is a very important parameter, as is the clinical exposure. So the story isn't this simple. And Diane is going to talk in detail on this later. But I wanted to highlight here that threefold, whilst very weak in vitro, could still be significant. Next slide, please. So here then is all of the in vitro data that we had gathered. And on the top panels, I'm showing the EC50, and on the bottom panels, I'm showing Emax, and I've highlighted that threefold from the previous slide. And the data here is again divided on the left-hand side panels for compounds that just had in vitro induction, and on the right-hand side, compounds that had induction and inhibition. And data came from at least 3 laboratories per compound, and each data point represents a different donor or in a different lab. And so immediately you can see there is a degree of variability in the in vitro data similar to what we saw in the clinical data. What was surprising to many of us is that this variability also exists in the EC50. We were reasonably understood that the Emax might be variable donor to donor because we understand there are donor differences. However, the dogma was that the EC50 should be more reasonably controlled. However, you can still see in this data set some variability there. And I want to underscore that despite this variability, we're still comfortable that we can use this data to make decisions about clinical DDI risk. And again, a nod to Diane, who will talk about that later. And so if we dig into this variability a little bit more on the next slide, we really, in the group, spent a lot of time thinking what could be causing this variability. Is it differences in induction protocol? Is it differences in how these experiments are run? And so we really focused on how we could understand this. And I think the data here describes that this is really variability that exists within donors. On the top panel, I'm showing the rifampicin EC50, and the bottom panel, it's the fold induction and with the Emax. And each of these sets of data, the letters represent a different donor, and each data point each donor is run in the same laboratory under the same experimental protocol, often by the same individuals. And you can see that the within donor variability is still significant even when protocol and lab is conserved. And so that intra-donor variability is almost as much as the within donor variability. And this, I think, is quite striking and something that we need to be aware of. So if we go to the next slide, we're really thinking what could be the causes of this variability. And the first thing that we observed is that it's probably the basal level that is variable, and that's what's driving the fold induction differences. And so what you can see here on the left is PCR data for a single donor that has been repeated over 60 times. And what we have is the delta CT values on the left and the fold induction on the right. And for folks who aren't used to looking at delta CT values, just remember that the lower the CT value means there's the more cDNA or the more gene signal that is present. And so the rifampicin or the induced state for these repeated experiments are the lower CT values in circles. And then the non-induced or the basal values are in the top of this graph on the left, and they're essentially shown in these squares and triangles. Now this data set is rank ordered by increasing baseline 3A4CT. And so essentially what that means is the lower the basal activity, the further to the right you are in this experiment number. And then when we look at the fold induction panel on the right, you can see that the further to the right you are, where the basal values are lower, trends to the higher fold induction. So perhaps not surprisingly, we understand that it's the basal variability that's driving the variability in induction. Okay, the other thing to note is, it's off the bottom of my screen, but I hope others can see it, is that similar observations were made in other donors, and in our manuscript we present that as supplemental material. We can go and the next slide. One of the questions that we asked ourselves was, okay, we're repeating this donor over many times. Is the variability due to the fact that this donor is being cryostored for many years potentially? Is that causing some of the variability? And the other question we asked was, well, how many times do we have to repeat a donor before we have an understanding of the extent of variability within that donor? Because there are differences donor to donor there. And so this panel shows a donor represented in chronological order. So this is the actual experiments as they were done in real time. And you can see for rifampicin point estimates that the variability that you're seeing in the first 10 experiments is pretty similar to the variability you're seeing towards the latter 40s or 50s. There's one spike where there's clearly an outlier in the fold induction, but the variability is not dependent on the order of, or the time or age of the donor. And so this does make you think, okay, if I want to assess what my donor is going to do, how many times do I need to repeat that donor before I understand? And I think it's certainly more than the 1 or 2 times that many of us might evaluate a donor before purchasing a big batch of cells. Okay, so I want to switch gears a little bit now and start to come back to more of the recommendations. And I want to show you some more of the data that we generated within the group. And this is rifampicin, 3A4 mRNA, just from single point control. So in these experiments, rifampicin was included as either 10 micromolar or 20 micromolar as the positive control. Each of these H's represents a different donor, and this is collected data from multiple laboratories. And what I'm showing on the axis here is fold induction. And you can see there is a range of rifampicin responses within each donor, and that variability is dependent on the actual donor itself. Some donors have less variability than others. And so when we're looking at this shows us the power, again, of a consortium. We have over close to 600 data points here. I don't think a single lab would really ever have this volume of data to understand what we're dealing with induction. And so based on this, we felt that if we want to qualify a compound as negative and we have this sort of variability in our positive control, we want to make sure that there's sufficient dynamic range. And so we're recommending that the rifampicin needs to be greater than 10-fold within the donor if you're using 2-fold threshold to qualify a negative compound. And the dotted line on this chart shows you the 6-fold. And you can see in some cases there would be experiments within a donor that would not meet this criteria of even 6-fold and certainly not 10-fold. And so the question you might be asking yourself is, well, why did you choose 10-fold? And to some degree, it was quite pragmatic. And we did some Monte Carlo simulations based on this data set, where we essentially then simulated out another 10,000 virtual iterations and asked the question, what's the probability that the rifampicin response is going to be greater than 6-fold, greater than 10-fold, or greater than 20-fold? And you can see here for greater than 10-fold, we're pretty comfortable that more than one donor would have a response for rifampicin greater than 10-fold, and that's showing like 98%. For about 84% that both of the donors of the 3 run would give you more than 10-fold. Now the 20-fold, the reason we didn't set the threshold that high is pragmatically you would likely have too many experiments failing if you wanted to qualify that. And we felt the 10-fold was probably reasonable. And importantly, this 10-fold threshold, the way we're thinking about this is that it's important only if you want to qualify your compound as a negative. So think back to that very weak induction data. We want to make sure that we can capture the 3 or the 4-fold induction with confidence. However, if you have a compound that's showing you a clear dose response, concentration response with an EC50 and an Emax, then that data is of value independent of where the rifampicin response is in that donor. So a little bit of speculation on the source of some of the induction variability. The first, obviously, the clinical variability that we showed. This is obviously due to individual variability, probably in CYP basal expressions in the gut and the liver. Obviously, then also the dietary impact, particularly in the gut, could play a role on that basal expression. And this may be why, for very similar apparent clinical study designs, you would actually get a different induction response. And there's something about the study design that also needs to be considered, such as when the doses are staggered, or making sure that you've achieved steady state. And the induction working group has a clinical commentary that hopefully will be coming out in the new year that really talks in greater detail to this. And when we think about in vitro, the variability that we've shown, and remember that the data I've already explained to you, our thoughts are that really this is subtle differences in the culture conditions that result in different unbound intracellular drug concentrations. And what might this really mean? Differences in seeding density and attachment efficiency for experiment to experiment might lead to different density or different quality of monolayers. And we know that the monolayer is very dependent on establishing a good basal level. That variability in the enzymes that is caused and the transporters obviously within the study could then result in different clearance or intracellular uptake for your individual compounds. This could also influence the albumin secretion, which can drive changes in the unbound fraction, and also subtle differences in dosing. We do large fold dilutions of our test article to make sure vehicle control levels are very low. And essentially there could be variations in the amount of drug you're dosing. And then also the temperature of that media could affect the differences in the cells themselves, and how long it takes an operator to change the media. All of these very subtle things could contribute to the variability we're seeing, even when you pull the exact same donor out of the freezer. Some speculation here. The data I've shown you is all mRNA data. It's possible this is a more variable endpoint. We don't have a lot of activity data, but the data that we do have is in the paper, and it is certainly appears to be less variable. However, it's a less useful endpoint also because of the prevalence of the time-dependent inhibition, meaning that an activity measurement is not going to be valuable. So a question you might be asking is there value in quantifying proteins? And I think we understand the Western blot semi-quant is not going to be helpful, but now that there are much more robust methods using LC-MS to determine protein levels, this may be a way of counteracting some of the variability. We can also ask the question, is there a need for a more robust system in vitro? And I wonder whether physiologically relevant systems that are starting to be used might reduce some of this variability because they would reduce the impact of these subtle differences in culture conditions. So let's come back then to some more of the data and the recommendations. And I'm going to talk about that twofold in a little more detail. To do that, first I have to introduce flunazanil, the negative control, in a little more detail. This is as a benzodiazepine receptor antagonist, and importantly, it's not an inducer of CYP3A4 mRNA or activity in vitro, and it's also not an inducer in the clinic, and has been used as such as a negative control in some in vitro induction studies. And the working group gathered a large data set of single point 25 micromolar flunazanil data from repeated hepatocyte donor experiments and evaluated essentially the signal to noise. So how many times would you get a positive induction threshold for a compound that we know shouldn't cause induction? And I'm going to show you that data now. And, oh, too many. So here's the fold induction for flunazanil in 10 different hepatocyte donors. And you can see the majority of data, over 300 of the 314, were within twofold, so between 0.5 and 2-fold induction. However, there were some incidences, and in half the donors, at least one of the repeated experiments did fall outside that twofold. And so there are some chances of some false positives for this negative control. And this is that same data just tabulated, and the donors, the 10 donors, the number of repeated experiments, the minimum, the maximum, and then the mean and standard deviation. And then here is the mean of all the donors. And we took a statistical approach to look at the limit of blank and the limit of detection. And essentially, these are the 95% confidence that you would have a true negative with a limited blank or a true positive with a limited detection. And these values shown here, 1.8 and 2.5, essentially bracket the standard twofold that we've already been using. And so we believe this data then supports that twofold. Sorry, I've lost the slide somehow. That we believe this data then supports twofold in that variability. Sorry. Mahalaya, can you help us get back to the slide set? Thank you. And what we're seeing here then is if twofold sits in the middle of what we believe is a true blank and a true negative and a true positive, there's no real value in changing the thresholds. We also did a series of simulations to explore false positive risk because nearly half those donors had at least one point where you would show close to threefold induction. And so again, we use Monte Carlo simulations to project out 10,000 iterations. And here we ask the question, what's the probability we would have in one of the three donors greater than a twofold response for a negative control? And you can see that the probability of seeing a false positive in all donors is very low, still low in more than two donors. However, there's an 8% chance that you might get one donor give you greater than twofold. And because of this, we're recommending that if you see twofold induction, you need to see that in more than one donor before you qualify your compound as an in vitro inducer. And this goes without saying, but I do want to remind this audience that just because you identify something as an in vitro inducer doesn't mean that you have a clinical DDI risk. So I don't think we need to be afraid of identifying induction. It's just a trigger for then going forwards and doing some risk assessment with mathematical modeling that Diane will take you through. And then you decide whether you would need to do a clinical DDI study. So now let's come back to that negative control. So one of the other recommendations is that it adds no value beyond the vehicle control. And paradoxically, it's actually been quite valuable for our induction working group because it helped us establish that twofold control was useful. And so when you're interrogating the large data set, it had a lot of value. But then in an individual experiment, when you're assessing your test compound, we felt that it really doesn't do anything beyond the control. And what we're showing here is, again, gene data. So this is the housekeeping gene, CT values. You can see that flunazenil and DMSO and rifampicin all track. So this is the no effect. And then if you look at the 3A4 mRNA CT, and again, the lower value means that there's more of that gene present. Flunazenil and DMSO essentially track very closely. And so really the negative control is not telling you anything, but your vehicle control is not. Okay, so switching gears to another recommendation from the working group, and this is around the normalization of your test article response to a positive control. And historically, as a field, we recognize this in vitro variability that we see in induction studies. And at the time, early days when we were all using fresh donors, it was clear this was donor-to-donor variability. I think we've shown in the data we've looked at this morning that this is variability that exists within a donor. And so the thoughts were, if you have a positive control, maybe you can normalize that variability that you see run to run. And what I'm showing here is rifampicin data in this first panel. And this is fold induction data from the literature. This was published by Zhang et al. And DMZ, and 3 different donors. And if rifampicin was able to calibrate, if you like, in vitro the response, if the rifampicin signal is high, your test article, you might also expect a higher induction. If your rifampicin signal was low, you might also expect a lower signal for your test article. And so therefore, the theory go that if you normalize, then you can reduce that variability lot to lot. However, if that were true, if we look at this rifampicin data here, this dashed line you see is the highest response if this was true, that your rifampicin response was going to show you your compound test response signal too, that dashed line would be the highest signal in all of the donors. And you can see that's not the case. In one donor it is, but in this other, for one compound it is, so sulfapyrazine. However, for another compound like carbamazepine, that is not the case. And so another way of looking at this data is looking at the percent of positive control. And this is an expanded data set where now I have normalized the data. And so rifampicin now exists as 100% here. And if this were normalizing the response, you would see that there would be little variability in the response within compounds. And for some compounds, that's true. If we look at omeprazole or this series of compounds, the response relative to rifampicin is pretty similar. However, for some compounds, that's not true. So again, sulfapyrazine, compound 11. Let's look at another, oxocarbazine. And so if you have a test compound, you don't know whether you're going to be one of the compounds that does normalize well or doesn't. And for that reason, in vitro, we're saying that normalizing doesn't seem to add a lot of benefit for you. And so I'm going to end here and hand over to Diane, but I want to summarize all the recommendations from this portion of the work. And again, we have two that support the guidances. Continue to use 3 donors because of the in vitro variability we see. Use twofold as a threshold for a positive identification for CYP3A4 mRNA. However, if you do see greater than twofold, make sure you see it in 2 donors to avoid false positive risk. If you are qualifying a compound as a negative, make sure you have sufficient dynamic range in your experiment, so rifampicin should be greater than 10-fold. Normalizing data to the percent positive control doesn't seem to control in vitro for the variability we see across experiments. And finally, adding a negative control isn't adding any value beyond the vehicle control. And I'll hand over to Diane now.
Thanks, Jane. So I'm going to focus on.
Excuse me, Diane. I'm going to interrupt for a second. We're cutting off the bottom of the slides, and we're going to see if Malia can adjust the size so that everyone can see all of the slide. Can we take just a second to try that?
Sure, certainly.
Maybe go forward to the next slide or a slide where we've got writing, just to make sure that we can see all of it.
Okay, I'm going to try to.
Even just go back to the last slide.
Yeah, I'm having the same problem. Oh, there it goes.
Yeah, so we're still cutting off the bottom.
Okay, let's just go ahead and we can circulate the slides at the conclusion. I know we've got the content. Yep.
Okay. All right, so thank you, Jane. The focus on the next portion of the presentation will be to go over the methods that we use to evaluate whether this extensive data set could be used to evaluate in vitro and vivo extrapolation. So can the in vitro data be used to predict the clinical risk? And as I'm sure we all know, in vitro parameters do not equal directly the clinical response. So I'm actually having the same problem as Jane in that I can't manually transition from my computer. So if you just want to advance through the full slide, that would be fine. So in order to context the in vitro clinical induction parameters against the clinical outcome, we need a number of clinical parameters, such as the dose, molecular weight, the steady state Cmax, absorption rate, fraction absorbed, plasma protein binding. Early in development, often a lot of these clinical parameters aren't necessarily available. So we're doing these risk assessments based on predicted human data. Additionally, we have availability of a number of approaches in terms of mathematical models to assess the potential for clinical risk. And these range from those that require minimal data, such as a simple Emax over EC50 rank order or binning approach, all the way to those very data-intensive, complicated models, such as PBPK. So for the purposes of our assessment within the induction working group, we actually didn't collect enough data to be able to apply PBPK as a method for assessing quantitative prediction. And the rest of the presentation will focus more on these more simplistic models, those that are both recommended by the regulatory guidances as well as up to the static mechanistic model. I'll try to, there we go. So just as a historical perspective, we have actually come a long way over the last almost 2 decades from the point where we really established the utility of the human hepatocyte model to be predictive of induction, to early 2000s, where we were really using binning ranking approaches based on the in vitro induction data, the recommendation to kind of use the prototypical inducer response as a reference for cutoff values in terms of clinical risk, to the mid 2000s, where we started really developing more sophisticated experimental models, including the calibration approaches that I'll talk to in a little bit more detail, the relative induction score approach, all the way to dynamic modeling, which is really coming of age now, and I think developing a capability to be quite a bit more quantitative in our predictions of clinical risk. I'd also like to just remind everybody that the induction working group has been really actively compiling and trying to develop recommendations around this data for the past 3 years. So we're really at a point where we're still kind of learning about the potential models that we can use to apply a clinical risk assessment, and emerging in terms of PBPK and the capabilities. Next slide, please. At the same time, the regulatory landscape is shifting. So when we started this analysis, we had guidance from the FDA and PMDA, as well as the EMA. There were some differences between FDA and PMDA compared to EMA, but in general, the PMDA and FDA were pretty consistent in the recommended approaches for modeling induction potential using basic models. These included the R calculation for the R3 value, where the cutoff was 0.9 using the total Cmax. And in terms of the EMA, as Jane has mentioned, the basic cutoff criteria is really a positive induction signal is that which is greater than 2-fold mRNA level at a concentration which is 50-fold the unbound Cmax, or the gut concentration, which would be calculated using 0.1 times dose over 250. During the manuscript review, we actually received draft updated guidances, and this was a very positive thing. I think it indicates that regulators are really taking the literature that we're generating seriously. A number of publications between 2012 and 2017 really highlighted that this was a very conservative cutoff. So they actually made some updates to the recommendations. If you advance to the next table. So now the FDA and PMDA, again, very well aligned, actually incorporated some additional basic model approaches to evaluating the clinical induction risk. They included this fold cutoff consistent with the EMA guideline, but also included calibration approaches such as the relative induction score. And then they also changed the cutoff criteria and input concentration for the R3 equation. Again, this is likely because of data indicating that the original equations were kind of over predicting and leading to a lot of false positives. They also mention in the new draft guidances some criteria related to the gut concentration. And there is a bit of difference between the EMA and PMDA and the FDA in this approach. So the FDA gut equation does not include this scalar of 0.1. If you go to the next slide, great. You can just click through all of the animations. That will make it easier. So this is just kind of a decision tree. And apologies again that the bottom of the slide is cut off, but this will be circulated after the presentation. So essentially, the decision tree between the FDA and PMDA, which is very well aligned, and the EMA, do have some differences in terms of how you would conduct a clinical risk assessment once you have generated the in vitro induction parameters. They both agree, or all three agree, that mRNA changes should be measured from 3 different human hepatocyte donors. As Jane has gone over, this is a recommendation that we are also making based on our data analysis. The FDA, as I mentioned in the previous table, has recommended some additional basic models for assessing the potential for clinical induction risk. And if any of these models exceed the proposed cutoff, then you can either go directly to conducting a clinical study, which is the box that's cut off at the bottom, or you can proceed with mechanistic models. So this would include a static mechanistic model or a dynamic or PBPK modeling. The EMA is a little bit different in that it uses the fold cutoff criteria that I've described previously. You can either move directly into a clinical study, or you can do some calibration approach or the mechanistic static model. The EMA, in contrast to FDA and the PMDA, does not include PBPK modeling as a potential method for excluding the need to do a clinical investigation when these basic models result in a positive. And both of them agree that the worst case donor should be used to assess clinical risk. So moving into an overview of how we collected the in vitro data, we initially requested that proprietary data be provided by the IQ member companies. This yielded 16 of the 51 compounds, so about 30% of our total data set. At the same time, we asked for the participating labs to provide in vitro data for standard inducers that they had historical data for within their labs. This was, again, approximately 30% of our data set. To build upon the data in terms of no clinical or mild clinical induction, we actually sourced some in vitro induction parameters from literature sources. This was a similar amount of data points as our proprietary compound data points. So overall, the majority of our data collection efforts really were driven by data that was provided anonymously through our survey efforts. Lastly, we wanted to build upon the data set where we see in vitro induction, but not clinical induction. So we identified 5 compounds where there was clinical DDI data available, but we were not able to find in vitro induction parameters, and we generated that within the induction working group from 5 different labs. So that actually accounted for 10% of the total data that we had available. If you go to the next slide. Thank you. So I'm going to show a number of data tables, and the intent really is not to go through each one individually. I just wanted to give everybody in the audience a sense of the amount of data that was collected, but also highlight that a number of compounds had far greater number of data points than others. So as an example, ritonavir, we had quite a bit of both in vitro data as well as clinical studies. This data table is showing where we see in vitro induction, but the net outcome is clinical inhibition. And interestingly, about 38% of the proprietary compounds that we collected, so those that are designated here with the compound number, actually yielded clinical inhibition rather than induction. The other thing I wanted to highlight, and I'll use this table as an example, is that in some cases, particularly with a proprietary compound, we have very limited data in terms of clinical outcome. And in some cases, such as with clotrimazole, we actually didn't have a sensitive CYP3A clinical DDI study. So this was part of the rationale for including clinical data where there was induction and there was some contribution of CYP3A, but wasn't necessarily a sensitive CYP3A substrate. So if you go to the next table, I'll go through this relatively quickly. Oh, I think maybe I can try. I can actually advance by clicking on the arrow. So I'll do that from now on. So this is the no induction data table. And as you can see, we were really able to populate this with a large number of compounds, again, with varying amounts of input in terms of both in vitro parameter data generation, as well as the number of clinical studies. And again, we have a number of compounds where we didn't actually have a sensitive CYP3A study to compare to, so we really relied more on the all substrate data. I'm gonna also point out where there are green stars. These are the compounds that we identified through the literature review as those where there was some clinical readout, but we didn't have in vitro induction parameters to evaluate IV-IVE. So members of the induction working group from 5 different labs generated these in vitro induction parameters. The clinical week induction, we also had a very large data set. And again, we were able to increase the number of compounds within this data set by generating in vitro induction parameters within the working group. Moderate inducers, actually quite a number of the moderate inducers, around 30% of the proprietary compounds that were collected turned out to be clinically moderate inducers. So this was really helpful in building out this data set and really understanding some of the IV-IVE for this type of clinical observation. And the clinical strong inducers, actually, there were really only 2 that were commonly, the clinical outcome was strong induction. Many of the moderate inducers could yield strong induction in some cases, but it wasn't every clinical study. Rifampicin, which we all use as a prototypical inducer, actually was one of our largest data sets, both for in vitro induction parameters as well as the number of clinical studies. And we also collected a proprietary compound where the clinical outcome was strong induction. So overall, we had a very large data set, a high number of inducers or non-inducers, in vitro inducers that didn't result in any clinically meaningful induction. A lot of in vitro parameters to use in our IV-IVE assessment, and a very large number of clinical studies to kind of reference our evaluation of IV-IVE. As I've already mentioned, we had a very strong data set for both no effect and weak induction. And this was intentional because we really wanted to try to figure out if we could adopt some best practices in terms of identifying these to avoid, hopefully, some of these false positive compounds. So before I go into some of the data analysis, I just wanted to review some of the complicating factors. So as Jane already mentioned, we actually had a large number of our data set where we see both in vitro induction and inhibition. And this can very much complicate the interpretation if we're only considering the induction component, which we are doing when we're using just mRNA as a readout and not considering the inhibition as we do for most of the basic models. As I mentioned, we also had quite a number of compounds that we collected where we didn't have CYP3A sensitive substrate data. So a quarter of all of the compounds, we were able to establish in vitro and vivo extrapolation by considering all of the substrates. One of the limitations of that approach, and we're going to go into this in a bit more detail in the clinical commentary that is forthcoming, is the selectivity of the substrate towards specific enzymes that may also be co-regulated, so also induced. This can significantly affect the clinical outcome. So if we just look at ritonavir as an example, ritonavir is a well-known time-dependent inhibitor of CYP3A. This, with sensitive CYP3A substrates, manifests in moderate to strong clinical inhibition. But if we use a probe such as voriconazole, which is metabolized by not only 3A, but also 2Cs, UGTs, which are also inducible, the net clinical outcome is actually strong induction. So this is something to keep in mind when you're designing clinical studies or trying to reference between clinical studies, where the selectivity of the substrate may be towards additional inducible enzymes in terms of the extent of metabolism. I also wanted to go through a little bit of the variability prior to going into kind of the workflow for assessing IVIVE. We actually use median for our assessment, and part of the reason for this is because, one, we have extensive variability both in vivo, so the bottom axis represents what was the observed AUCR, and you can see rifampicin here, which is being highlighted, has quite an extent of variability in terms of clinical outcome. Additionally, there's quite a bit of variability in the in vitro data point, as Jane has already kind of gone into in detail. If we look at the distribution, what I'm showing here is of rifampicin, but the data doesn't tend to be normally distributed. So the median, using the median value actually is a bit better than mean because it's not susceptible to these outliers, which is why we sought to evaluate the median induction parameters generated in vitro as well as the median clinical data points, because we didn't really want to bias our interpretation based on some of these outlier data. So I'm just going to quickly remind everyone of the data analysis paradigm. So we had all of this in vitro data. We generated both the median induction parameters, as I described. We also looked at the worst case induction parameters. So those parameters where the EC50 was the lowest and the Emax was the highest. We referenced the ability of these parameters to be useful in IVIVE based on binning, and we evaluated them against the median clinical induction or the worst case clinical induction. So we evaluated a number of equations against a number of reference points. We had quite a bit of data to sift through based on this paradigm. Because of that, what I'm going to actually focus on for the rest of this talk is just the median in vitro induction parameters compared to the median clinical induction data. All of the data analysis is presented in the paper if you're interested in some of these other comparisons. So we also looked at a number of mathematical models, which I'm showing here. We have various input parameters for the different recommended equations based on the regulatory guidance documents. Because of the shifting landscape with the FDA and PMDA, the draft guidance being coming out in 2017, we also included some variations of those equations as well. We gauged the binning potential based on whether we were accurately able to identify true positives and true negatives and avoid false negatives and false positives. We had a secondary approach where we wanted to see whether these any of these methods were more predictive in terms of quantitative assessment. And we measured that based on those that were within twofold of the observed versus those which were within the bioequivalence. And I'm going to focus really a little bit more in detail on how we applied these calibration approaches, how we determined a scaling factor based on the large data set that we compiled within the working group. So the relative induction score approach has been successfully, at least in literature, successfully able to predict pretty quantitatively the extent of clinical response. And we wanted to evaluate this, but we didn't have the luxury of having what is recommended in the case of a single hepatocyte lot that has been rigorously validated in terms of the number of clinical inducers, the range of clinical inducers. We actually did request in one of our surveys for relative induction score data if it was available, and we didn't receive any. So the approach that we took from the data set that we collected was really similar to what was done for CYP2B6 recently with another IQ working group, where all of the in vitro data was from various labs was used to construct a relative induction score curve. So the limitation here is that it's not a single donor that's been validated. And one of the limitations within our data set is that we didn't have a test set to compare to. But in general, this actually tended to be pretty accurate in terms of prediction capability. And as you can see, the confidence intervals are definitely reasonable. So that's the approach that we took in assessing our data set. What I'm showing on the left-hand side is the relative induction score curve, which was generated from using all of the in vitro data that was collected, and the median clinical response. So you can see by the smears of black dots that there's quite a bit of variability in the in vitro parameters that were generated. Again, we were able to fit the curve and get reasonable confidence intervals to establish the equation for evaluating its utility. We established a detection limit for the relative induction score of -20% for AUC change. And we generated similar data for using the slope with a detection limit which was not as low as that for the AUC change, but still reasonable in terms of fitting. Not surprisingly, we do see a pretty significant false positive rate. The relative induction score approach only considers induction. And as we've mentioned, we have a majority of the compounds collected have competing mechanisms of DDI. We also have a little bit of a false negative rate for the relative induction score approach. These false negatives were pleconaril and compound 15, which I'm going to go into a little bit more detail on why they could be false negatives. In general, the predictive performance was acceptable, but we're still not there in terms of our positive predictive performance, as well as the negative predictive performance, because we do have quite a high false positive rate. We also wanted to evaluate whether we could determine the scaling factor. So in the R3 equation, there's this D value, which is a scalar to kind of reduce the amount of false positives. There's been a number of publications recently where this scalar has been derived for individual hepatocyte lots, which is the preference in terms of establishing this within individual companies. But if we just consider that the in vitro data directly can be translated to a clinical outcome, so the scalar is set to 1, you can see that we're able to appropriately bin a lot of these as true positives. We have a pretty high incidence of false positive. We do have one false negative, and if you look at like the quantitative prediction, so these solid lines which represent 2 folds, we have quite a number of positive compounds that are designated as true positives, but are really over-predicted in terms of quantitative prediction. So we really wanted to identify which D value could reduce this distance here. but maintain the lower number of false negative values. In establishing this, we identified that a scalar of 0.3 would be able to reduce the number of false positives while not really impacting too extensively the number of false negatives. And it also brought a lot of these true positives back into like a more quantitative range. The numbers are displayed in the table here. So as you can see, this scalar did improve the false positive rate. There was an increased incidence in false negative of one compound, but the predictive performance was improved by applying a scalar. What I'm showing here is the table that is actually in the paper that gives you an idea of using the median values, how each of these equations performs. And if we just consider, the false positive rate, as I've walked you through some of the calibration approaches, we do tend to see quite a bit of high false positives. And this is something that I think is stemming from the idea of limiting the number of false negatives. So I want to go into a little bit more detail about the false negatives that we're observing and the potential for why. So we do see dexamethasone quite frequently as a false negative. And we had some discussions within the induction working group, and a lot of us think because we do add this as an additive to the culture medium to maintain functionality of the hepatocyte system, we're underestimating these induction parameters. And that could be leading to a false negative. We have a similar interpretation of pleconaril where Jane went into a little bit of detail. It's definitely a weak in vitro inducer, but recent publication by the Vertex group has shown that it actually in the media is depleted quite quickly. This could be due to nonspecific binding, and therefore, because we're using nominal concentrations to estimate induction parameters, we may be underestimating these, and that could also lead to a false negative. Another thought is this thought around protein binding. So when we're considering highly protein-bound drugs, the FDA guidance recommends that we don't use a value greater than 99%. Is that impacting this, where we have values reported that are above or greater than 99.9% binding? Additionally, some of the proprietary compounds that we have very limited in vitro data. So we may not have the most appropriate or best interpretation of the in vitro induction parameters based on this. So just going into a little bit more detail about the plasma protein binding, we did, out of the 51 compounds, have 7 that were reported to be greater than 99%. This is a 14% overall amount based on the entire data set, and those are all here in this table. 4 of the 16 proprietary compounds, or a quarter of them, were actually considered to be very highly protein bound. What's interesting is that even though these top 5 compounds reported very high binding, and these values were used in the assessment for IVIVE, they actually gave us correct interpretation of results. This compound 13 was a false positive, so the binding really didn't have any impact on the interpretation of results. However, when we considered the cutoff of 0.01 for compound 15, we were able to generate an accurate binning. So this does show that plasma protein binding should be considered in part of the in vitro and vivo extrapolation. But if we look at the whole data set, a lot of these false negatives, those that are highlighted in blue, actually don't have very high plasma protein binding at all. So it's not the only thing to consider in terms of where our false negatives may be coming from. As an example, if we look at oxcarbazepine, which is very, very low plasma protein binding, this comes up as a false positive, or false negative rather, in in the equations where we're even accounting or limiting it to the plasma protein binding. And also, when we consider the quantitative predictions, our average steady state unbound concentration actually gave us the best quantitative prediction, but it also led to the majority of false negative compounds. And again, if we consider our proprietary compounds where there was a false negative using the unbound concentration, using this scaling approach actually eliminated all of those false negative compounds except for 15, which I just kind of spoke about. But it didn't eliminate the pleconazole or dexamethasone as false negative. So again, there's more that needs to be considered in terms of the in vitro and vivo extrapolation and elimination of false negatives than just the protein binding. So in summary, we were able to evaluate quantitative prediction. We could use the in vitro data to assess clinical risk, but really it's a situational preference in terms of what equations or methodologies would be used between companies. And we do see an improvement in the quantitative prediction when the average concentration is used. And again, this is really just excuse me, something that within company needs to be considered when evaluating the potential for clinical induction risk based on the in vitro induction data. So just some quick key takeaways from this assessment. We did establish that in vitro data could be used to conduct a clinical risk assessment despite the variability that is observed. We do see a high false positive rate for the majority of these basic equations. When we use the median induction data rather than worst case data, we are able to reduce the false positive rate and improve quantitative prediction. If you use a cutoff of 30 times unbound steady state Cmax rather than 50, you can improve the false positive rate. We established a cutoff for the dose level using the EMA basic equation when evaluating gut induction. When you can't establish in vitro induction parameters, either due to solubility or cytotoxicity, both the F2, which is that concentration leading to twofold induction, as well as the slope value, are useful for estimating clinical risk. And we have data that indicates that the average concentration really provides the best quantitative estimate. So this really highlights the potential that we have in developing PBPK. While it was out of scope for this analysis, it should continue to be explored. I'm not going to go over this slide. This is just the recommendations that were made in the recently published white paper. Quick review of what the induction working group has accomplished to this point. We have 2 white papers. We've conducted these 2 webinars, and we have 2 papers that are planned for early next year. With that, I would like to thank all of the members of the induction working group team. As you can see, we have experts from across industry, as well as some expert statisticians. And with that, we will thank you for your attention and turn it over to any questions you may have.
Thanks, Jane and Diane. So please submit questions online. We have a few. I'm going to start, and then Naresh is going to take over. So the first question is kind of a benchmarking question, which is, how do these results compare to other publications on CYP3A-mediated induction and in vitro and vivo extrapolation?
So Donald, perhaps I can take the in vitro aspect and Diane can talk about the extrapolation. I think our data here from the in vitro aspect is probably one of the largest data sets that's been pulled together, and it's allowed us to see things that were previously not really well understood. And so I think that really helps us understand both the clinical and the in vitro variability and understand what we could be doing when we're interpreting this data and the best practices. And for IVIVE, I'll turn that to Diane.
Thanks, Jane. So I agree that at this extensive data set really enabled us to look into further detail some areas where there may be gaps, such as those non-clinically relevant inducers or weak inducers. So I think we're really just building upon the literature that's already available. But in general, the IVIVE is definitely in agreement with those publications. The more sophisticated our models get, the more seemingly capable we are of quantitative predictions. So while the basic models definitely serve to identify those potential positive clinical inducers, it's definitely advantageous to move forward with as in the development line with additional investigations using more sophisticated models such as PBPK.
Great, thanks. So one of the topics that came up in the first webinar was cytotoxicity and our ability to measure that. And so the question is, how do you rule out cytotoxicity as a limiting factor for Emacs when cytotoxicity assays are notoriously insensitive?
Yeah, I think that's a great question, and it's something you can understand the Induction Working Group has spent quite a lot of time discussing. And we agree a lot of the very standard cytotox assays are insensitive, and I think people are thinking particularly of the LDH or lactate dehydrogenase leakage assay, which is really only going to pick up very gross cell death when you've ruptured your membranes and your cells are leaking. However, we have more than one tool at our disposal, and within the induction group, we did use as many different markers of cytotoxicity as we could to, for want of a better word, clean up the data. And these include an assessment of the housekeeping gene. I think if you're starting to see decreasing cell numbers, your housekeeping gene can plummet. Morphology really is one of your best markers of cell health. That can be somewhat subjective, so you do need somebody who is very well trained in assessing cell health, but that really is the first indicator. There are also other assays that assess mitochondrial function that don't mean you have to sacrifice your cells. So you can get a mitochondrial function assessment using kind of Alamar Blue or Presto Blue assays, for example, that really are a little more sensitive than your LDH leakage assays. And so we personally, I would look at multiple different endpoints, and if any one of those showed me cytotoxicity, then I would exclude data because it could be unreliable. What was interesting is we had a good debate on this at one of the sessions at the ISSX for the Hepatocyte Researchers Association, and I think this really is something that the field is grappling with, and I'd invite others on the team to add their thoughts now.
Great. Well, I'll ask one more, and then I'll turn it over to Naresh. And this is a couple of questions related to the recommendation of a 10-fold rifampicin response to qualify a donor, particularly for a negative response. Is there a fold induction that would be too high? So is there a window here that we should be thinking about?
Yeah, that's another great question, and again, something we've grappled with. And I think it's going to be situational and donor dependent. I think when we're thinking about the importance of qualifying positive or negative for a test article, then I think that the floor as opposed to the ceiling is really important. That's where that 10-fold came. We couldn't get agreement on what to recommend would be too high for an induction. I think people understand that if you see very high fold induction, that's indicative of a low basal activity, which could be a marker that your cell culture is not optimal. But it's also actually donor dependent. Some donors do respond and are more sensitive. And so where do you draw that line? Do you draw it at 50-fold? Do you draw it at 100-fold? And I think the experience differs within organizations. And so we wanted to highlight that, and in the manuscript we do call it out, but we weren't comfortable setting an upper limit.
Great.
Donald, I can take over from here.
Thank you.
So another question to Jane and Diane. You did on several occasions mention variability or variability in data of when conducting in vitro studies. Can you speculate on the causes for clinical induction variability?
Thanks, Nash. I'll try to take this one. So Jane did highlight a couple of potential contributors to the clinical response or outcome, and that really did focus on kind of the basal variability that's observed in the population for CYP3A, and that's typically measured with like oral midazolam or even some of these endogenous biomarkers. So that definitely could be a contributing factor. In terms of the data collected within the working group, we also have the complicating factor of substrates, which may also be metabolized by other inducible enzymes. So just looking at CYP3A may not account for this. There's also the potential for this to lead to different effects when there's time-dependent or competitive inhibition of CYP3A observed. So those are just a handful of potential contributors. And as we've mentioned a couple times, we'll have the clinical commentary come out where we really try to delve into this aspect a little bit deeper.
Thanks, Diane. Another question, for the most part, pharmaceutical companies, CRO, use monoculture sandwich culture systems to assess induction risk, in vitro induction risk. Is the IWG looking into other in vitro systems?
I think that's a great question, and you could imagine within the induction working group there's been a lot of effort, but we are focused upon what the gold standard is right now. In the short term, there's not a plan to actually start generating data within the induction working group around this, but personally I think it is an area that we should be exploring as a field. And so I would hope in the future, maybe some future efforts could be to explore other systems here. And Naresh, I don't know what your thoughts are on this too.
Yeah, so I can add my 2 cents as well. I mean, we have looked into other systems, including cold culture systems, etc. One of the advantages we saw with using that type of system is the retention of transporter activity. So we were dealing with a project where, in addition to compounds being inducers, there were also substrates of uptake transporters. So we wanted to use a system that was more quantitative, would give us a more quantitative assessment of induction risk. So we saw some benefits, but again, I think there needs to be more data generated by other companies to have any sort of additional confidence in these other systems. So another question, for the most part, the webinar focused on CYP3A4. Was data collected for other inducible enzymes? And is the IWG planning on putting that data out into the public domain?
I can take that one. We were hoping when we were gathering the clinical data that we would bring in some clinical induction for particularly 1A2 and 2B6 so that we could focus on the translatability. Unfortunately, we didn't add any vast amounts of data. I think there was only one study that came in that had a 1A2 response. And so from the clinical aspects, we can't do the same analysis as we've done for 3A4 mRNA. But from the in vitro aspect, we did gather 1A2 and 2B6 mRNA data. And yes, we would look to do the same sort of analysis around the thresholds, because it might be that they're different for those different enzymes, and that's something that is ongoing.
Thanks, Jane. Another question is regarding the use of biomarkers. So, for example, 4-hydroxycholesterol. Any comment related to that for assessing induction risk of NCEs?
Diane, do you want to take that one?
Sure, I can take that. So there is definitely some literature. There was actually a working group on this. So there's a publication related to the utility of some of these endogenous biomarkers for predicting drug-drug interaction. There's some conflicting literature in terms of the capability of the prediction potential. So again, this is something we cover in pretty extensive detail in the clinical commentary. I don't know that we have enough time here to go through individual examples, but I think biomarker data obviously would be very something that could be generated or assessed during a clinical study. So ideally, it's something that should be moved forward in terms of being able to reference, and even just in response to the inter-individual variability. It would definitely be helpful in trying to understand some of these differences in clinical response. So I think it's still emerging in terms of how predictive it may be, but it's apparently seemingly helpful in terms of interpretation.
Excellent. All right, we're right at 12:30. I would like to take this opportunity to thank Jane and Diane for giving this really interesting, insightful webinar. It's been a lot of work, lots of long hours, evenings, holidays, over the past 3-odd years. So again, thanks to both of you. To those of you who are listening, as was mentioned by Donald, we're open to feedback. Please do not hesitate to email any questions directly to the organizer, and we'll certainly try and respond to those. Thank you to everybody.
Thank you all.